All right, everybody, we're going to get started. Before we start lecture, I did want to bring up a resource that some of you may or may not know about, but this is, this is on the tech website. This is Jenny Ware, and Jenny Ware is a retention specialist for the College of Applied and Natural Sciences. So most of you are nursing majors. The rest of you are mostly somewhere in the College of Applied Natural Sciences. There may be a couple of you that are not, but there is also a retention specialist for you. But Jenny Ware is the retention specialist for the College of Applied and Natural Sciences. And she is an absolutely great resource if this, like many people in this class, feel a lot of times my class is the first time somebody really has to try. Okay, and it's something that's kind of hard. I know in high school it's really easy. A lot of it's very intuitive. And a lot of people find that they don't have to study and they do really well. And then they come to college and this is kind of the first class that they actually have to work at. And learning how to study is a skill and it's very difficult for some people. And it's nothing to be frustrated or ashamed about. It's very true for a lot of people. Learning how to study is absolutely a skill and it requires practice and learning different procedures. So she is really great for a lot of different reasons. Academic performance is one of them, but she's also really good at connecting people to the resources they need. She's a great resource for figuring out where you need to go. And she's always open to talk to anybody. So there's her email address right there. This is also on the LA Tech website. If you are having a hard time studying or you didn't do as well on your first exam as you would like or anything else, I really recommend that you reach out to Jenny. She is really, really helpful. A lot of students have said that her help really helped them learn how to study, and that applies to all classes, not just mine. So I really recommend talking to her if you're having any issues like that. She's really nice and kind and open, and she's really easy to talk to. So, yeah, there it is. Again, there's her email at the bottom, but this is also on the tech website. And I know she also hosts, like, in addition to doing, like, you can meet her for one-on-one meetings, but I know she also does, like, workshops on, like, note-taking and how to study and things like that, if you're having a hard time with that. So that is my recommendation to you. And she is also familiar with my procedures in my class. She has helped people with my class specifically before. So she has a little bit of experience with that. She knows how my class works. So she's a great resource. All right. So we are going to hop over to our next chapter and get started on our lecture. So today we start talking about microbial metabolism. And what metabolism is we've used that word kind of a lot, and you've probably heard that word around, okay? But what metabolism is in, so many words, it's the chemical reactions and the physical workings of a cell. So it's what a cell does to be alive and to be a cell. So that's everything from consuming nutrients to reproduction and replication. It's building molecules like fats and carbs and sugars. It's, if it's a plant, it's building flowers or fruits or whatever it is that an organism needs to be alive and reproduce. And that's a lot of things, right? Metabolism encompasses a lot of things. But again, it's just everything that a cell does. The most basic form of metabolisms that most humans can relate to is the breaking down of food so that we have energy, right? That's a form of metabolism. Our body, we eat food, our stomach and the rest of our digestive tract digests it and breaks down all that food so that we can use the components, right? We use the energy that we get from the food. We use the proteins and the sugars and the fats, the vitamins, the micronutrients, all of those things. Our body harnesses those and uses them to move our muscles and make our brain work and make our livers work and make our ovaries work, right? Everything that works in our body happens because of that initial digestion, Okay? And it's the same thing for unicellular organisms like bacteria. Now, they're not doing quite as many things, right? Bacteria don't have ovaries or kidneys or hearts or anything like that, but their processes still need to be pushed and created by something, okay? They still require energy. So metabolism, kind of at its simplest form, is the combination of two different processes, anabolism and catabolism. And anabolism is the building of large molecules from smaller parts, right? It's the building of our Lego tower that we've been talking about when we talked about carbs and lipids and fats. It's the physical process of building our Lego tower. That requires energy. And then the opposite end of that spectrum is catabolism. It's the breaking down of those large molecules into smaller building blocks, okay? And that is digestion. So other words that you could use to describe these would be synthesis, okay? Anabolism is the same thing as synthesis. It's creating something new. It's the putting together of the building blocks to make a bigger structure. And then catabolism is digestion, or the breaking down of large components. So again, kind of relating it back to how humans work, because that's how most of us can relate to this kind of stuff. When you eat food, what happens is your body breaks it down into its base components, right? You don't absorb an entire cheeseburger across your stomach lining, right? That's not how that works. You eat a cheeseburger and your stomach churns it up, and then it sends it to your small intestine where it churns it up even more, adds a bunch of acids and enzymes and things like that that break it down. And then it goes into your large intestine where those individual components, so individual sugars, one sugar at a time, one fat at a time, one amino acid at a time, get absorbed across your digestive tract. And then your body rebuilds those molecules from the building blocks. So for the most part, your body does not absorb whole molecules. Your body does not absorb starch, which is a long chain of glucoses, right? What your body does is it breaks that starch into individual glucose molecules and then absorbs them. When your body consumes protein, you don't absorb an entire protein across your digestive tract. Your body breaks it down into the individual amino acids and then absorbs the individual amino acids. And then once those things are inside your body, your organs put it all back together. That's a big part of what your liver does. Your liver puts all of that back together and makes those molecules again because it needs to be the stuff that we need. The protein I need is very different than the protein a pea needs, or a cow, or something else. I'm a different organism. So my body builds the proteins that I need from the building blocks that I give it. Okay? So anabolism, again, is the building. And the building is always going to require energy. Right? The building of that Lego tower, the picking up of the piece and stacking it and putting it on top of each other, that requires my arm to move, right? That requires some amount of energy input. Anabolic reactions require energy. Well, how do we get that energy? Our body gets that energy from those catabolic reactions. Catabolic reactions release energy. So when you digest food, and we're going to talk about how that works when we talk about ATP in just a couple of slides, but the way that your body works, or any other type of organism works, is when we break down those big molecules, when we digest them, that releases energy that our body can then use to do other things. Okay, so a lot of times these reactions are paired together. So a catabolic reaction releases energy that then makes this anabolic reaction happen. Okay, and then the anabolic reaction creates these larger molecules that can then be broken down again to create more energy, right? It's a cycle. And we're going to talk about ATP and how this cycle works more in just a minute. But that's really all metabolism is, at its very base definition. Metabolism is just the breaking down, building up and breaking down of molecules to release energy over and over and over again. Okay, and this is just another chart that shows kind of the same thing, is that catabolism is going to yield energy, right? So breaking down this glucose molecule into its base components, you don't need to memorize the names of all these components, but just know that breaking down this glucose in catabolism into its base precursor molecules is going to yield energy. And then the building up of those precursor molecules into building blocks, and then into macromolecules, and then into an entire cell are going to require the input of energy. They're going to use energy. Okay. All right, so one way that metabolic reactions happen, a big part of metabolism, is something called catalysts and enzymes. So catalysts are molecules that lower the amount of energy required to make a reaction happen. So any reaction that could happen, right, this is just a general thing, anything that could happen, like we said, requires some amount of energy, right? Those anabolic reactions require energy input. Catalysts lower the amount of energy required, okay? So catalysts, the thing that's really important to remember is when we're talking about a reaction, the substrates are what goes into a reaction. So these are some new words, but substrates is what it starts out as, the beginning of the reaction, and the products is what comes out. Okay, substrates and products. It's what you put into the machine and what comes out. And every reaction is going to require some type of energy, some amount of energy. The amount required is going to differ, but it's going to require some amount of energy. And that amount of energy is called the activation energy. Okay? The activation energy is just the energy required to make a reaction happen. That's the activation energy. Now, what a catalyst does is a catalyst is not going to change the substrates. It's not going to change the products. It's not going to change anything about the reaction other than the amount of energy it requires, okay? So the amount of energy without an enzyme is this much, right? This is just kind of a random number. But when you add an enzyme, it decreases the amount of energy required. Okay, so that usually is going to make a reaction happen faster. Okay, so another thing, it makes reactions happen more quickly. Now, I don't want you to think, the reason I kind of hesitated to put that up is that enzymes do not actually speed up a reaction. It's kind of like if we were talking about running a race, okay? Say we're running a race. You have two people that are running a race, and they are both running at exactly the same speed, okay? But if one person is running 50 meters and one person is running 100 meters, who's going to finish first? The person running 50 meters, right? Because they're running a shorter distance. It requires less energy from them. Both the runners are running the same speed. We're not changing how fast they're running. We're not changing what they're doing. The process of running is still exactly the same. All we're doing is we're making one person require less energy. Okay, they're just not going as far. That's kind of what that means. Okay, so the only thing catalysts do is lower the amount of energy required to make a reaction happen. Now, in real life, what this actually means is that a lot of times reactions won't happen if an enzyme is not present. Okay. If an enzyme is not, or a catalyst is not present, a lot of times in biological systems, a reaction won't actually happen because the amount of energy required to make that happen is too high. It just requires too much energy and our body cannot do it. So most of the time within biological systems, cells, humans, anything else, most of the time if a catalyst for a reaction is not there, the reaction will not happen. Okay. And so we're going to study a lot of different uses for catalysts and the things that they do. And hopefully this will make a little bit more sense as we talk more about it. Now an enzyme, I've used that word quite a few times. An enzyme is a protein catalyst. So catalysts can be anything, even something like heat is a catalyst, right? You heat something up, a lot of times it'll happen more quickly. Heat can be a catalyst, certain chemicals can be a catalyst, but most of the time in biological systems, so inside cells, catalysts are made out of proteins, and we call them enzymes. So an enzyme is just a protein catalyst. Now, something to keep in mind, which I think makes it a little bit easier, is that enzymes have a specific way of being named inside the human body. And it's actually pretty intuitive what an enzyme does when you look at its name. So most of the time, enzymes are named by the reaction that they catalyze plus the suffix ase. So what I mean by that, so if you see a word that has an ase on the end, it's probably a catalyst. It's probably an enzyme. So for example, one enzyme that we have is lipase. Okay, ase is our suffix. Lip, that prefix, where else do you see that kind of prefix? What other molecule did we talk about? Lipids. Okay, lipids start with lip. So a lipase breaks down lipids. Okay, a carbohydrate breaks down carbohydrates. All right, lactase breaks down lactose. So if you look at the word, a lot of time it is going to tell you what that enzyme does, okay, and which reaction that it changes or catalyzes. Now, most enzymes have what we call a lock and key design. What that kind of means in practice is that enzymes are designed to catalyze a specific reaction. Okay, they are a specific shape that is specifically designed. How many times can I say specific? They're a specific shape that are specifically designed to catalyze a specific reaction. All right, remember when we talked about proteins kind of briefly in chapter 1, I think? One of the things that I said is that when it comes to protein, the structure of the protein indicates the function of the protein. And this is a big reason why. The structure, the three-dimensional structure of a protein is going to be really important to the reaction that it catalyzes. Okay? And enzymes can do both break things down and put things together. Those are different types of reactions. So in this case right here, you have this substrate that is a red thing and a yellow thing stuck together. It binds to the enzyme, and that enzyme pulls them apart. Okay, and you end up with two products. But enzymes can also take two substrates and shove them together into one product. Okay, it can go either way, depending on the enzyme. Now notice, these are two different enzymes. These enzymes do two different things. And in most cases, enzymes catalyze one single reaction. So inside of a human body, we have millions of different enzymes that do millions of different things for us, because every single thing that happens in our body can be broken down into individual reactions. So every time you breathe, every time you move your arm, every time you have a thought, your body is using dozens and dozens and dozens of enzymes to break things up or put things together. Okay, it's a delicate balance of harnessing energy to create other things. Every movement of your body, every thought that you have is the result of building up or breaking down molecules. Everything your body does. And all of that applies to bacterial cells as well. Everything that they do can be broken down into individual reactions. And most of those individual reactions have individual enzymes that make them happen. So kind of back when I said that proteins, again, back in chapter 1 when we briefly talked about proteins, I said that it was really hard to overstate how important proteins were, and they kind of do everything. And this is why, because proteins are enzymes, and without enzymes, nothing else happens. Everything your body does is controlled by enzymes. Okay? And that is why a lot of genetic diseases that humans have are the result of a loss of an enzyme. Okay? And a lot of times just one enzyme is enough to affect an entire human being. So for example, have you guys ever heard of cystic fibrosis? Right? Most people have. Cystic fibrosis is a pretty debilitating genetic illness. Cystic fibrosis is the result of the loss of one single enzyme. It's actually not even the loss of one enzyme, it's the misformation of one enzyme. One enzyme inside of a human is misshapen. It doesn't have the right shape, so it can't catalyze a reaction. And so most people who have cystic fibrosis have a pretty debilitating life state, right? Their lungs don't work correctly, their pancreases don't work correctly, their digestive system doesn't work correctly. Most of them require lung transplants in their early teen years. Okay? Most of them get debilitating infections most of their entire life. Right? And there are a lot of other enzyme deficiencies. Something like Tay-Sachs disease is the loss of one protein. It's one protein, one enzyme, and it usually causes the death. Most of the time, if a fetus has Tay-Sachs, they are not born at all. But in the rare case that a person is born, they die usually before they're 2 years old. Okay? It's a devastating and debilitating illness. So enzymes cannot be understated. They're incredibly important. And a lot of people actually experience an enzyme deficiency that is a lot less debilitating than that, but it's more common. So this enzyme, or this is the enzyme down here, is lactase, right? And lactase is an enzyme that we humans have in our digestive tract that is meant to break down lactose. And lactose is a sugar. Where do you find lactose? Milk products, right? Milk and cheese, any kind of dairy products. It's a sugar that's found in milk products. If you don't have enough lactase in your body does not break down lactose, which will result in not lactose deficiency, lactose intolerance, couldn't think of the word, but it results in lactose intolerance. Okay, your body cannot break down the lactose that you consume, and what happens is that lactose is actually sent to the bacteria in your large intestine, who then digest it and create lots of gas that's painful and not very fun for anybody. Okay, so lactose intolerance is a form of an enzyme deficiency. So when you don't have enough lactase, your body cannot break down lactose. And so like all those pills, like dairy pill and whatever else, there's all those pills that you can take, Lactaid. Those are just lactase pills. So you take them when you consume dairy to help your body break it down. All right? And there are some other enzymes. This is another common one. It's called amylase. And amylase breaks down starches. We humans produce amylase inside of our mouths. It's a large component of our spit. So when you eat anything that has starch in it, like corn or potatoes or most other fruits, or vegetables, your spit or your saliva is going to start breaking down that food as soon as it enters your mouth. Now, those are two very small examples. But again, it's important to keep in mind that enzymes control everything, right? These are small examples, right? If you don't have enough lactase, then you get cramps and gassy when you eat too much milk, right? That's not that big of a deal. But enzymes control everything your body does. So losing certain types of enzymes can be devastating. Make sure I said everything I wanted to. Some other thing to keep in mind about enzymes is enzymes, for the most part, all enzymes are multi-use. What I mean by that is you can have a handful of enzymes inside of a cell, and that enzyme, the substrates will enter, the reaction will happen, the substrates detach, and then the enzyme does it again, over and over and over again, just like you see here. You have the enzyme by itself, the enzyme binds to its substrates, the reaction happens, and then the products detach, and the enzyme does it again. Okay? So enzymes, there doesn't need to be that many of them, and they don't need to be constantly produced. They are multi-use. All right. So now we have to talk about, in order to carry out these metabolic processes, cells do need some amount of energy. It doesn't matter if you have a catalyst. The catalyst decreases the amount of energy, but it doesn't make it go away. All reactions require, or all anabolic reactions require, some type of energy input. And we talked a little bit about this when we talked about metabolic diversity. We talked about chemoheterotrophs and autoheterotrophs and all that fun stuff, or photoheterotrophs. But where an organism gets its energy from is going to vary wildly. Okay, we talked about that before. They can get them anywhere from things like sugars, which is what's most common, or sunlight, or metals, or electricity, right? There are some organisms that get their energy from electricity, some that harvest it from iron or magnesium, all kinds of different things. Gold, right? There's an organism that exists for every type of energy source. But most of the organisms, just about everything we are going to talk about, obtains their energy from certain types of sugars. Okay, so that's what we're going to talk about the most. But it is important to just keep in the back of your mind that those catabolic reactions that produce energy can be from anything. But we're mostly talking about sugars. In particular, we're going to talk about glucose. Though there are lots of organisms, there are certain bacteria that only consume lactose, or only consume maltose, or only consume all these other sugars. But we're mostly going to be talking about glucose. All right. So something that's kind of important to keep in mind is that energy is not created in cells. Energy is not created, right? And that's one of those basic laws of thermodynamics, right? If you ever learned about this in chemistry. One of the basic laws of thermodynamics is energy is never created or destroyed. It is harvested and moved. It is not created, okay? It's whether or not you're able to harvest that energy source. For example, the sun, right? The sun produces energy all the time. When you stand out in the sun, when I stand out in the sun as a human being, I can feel the energy on my face. If I stand out in the sun long enough, that energy is going to burn my skin, right? And I'll get a sunburn. However, me as a human, I'm not capable of harvesting that energy. I can't turn it into energy that I can use. But the energy is still there. My body doesn't create energy. Neither does a plant, neither does any type of bacterial cell. They harvest it. They pull it from whatever source they are able to. In a similar way that a windmill doesn't create energy. Windmills don't create energy, they harvest it from the wind. They harvest the kinetic energy that a wind creates when it turns the big turbine. Okay, so cells never create energy. They harvest it or extract it and use it to fuel other reactions. And there's two types of reactions which are kind of similar to catabolic and anabolic, but not quite, is endergonic reactions and exergonic reactions. And we kind of talked about this before, that prefix endo means in, N-in, N-in. Endo means in. So endergonic reactions require energy to come in. They're reactions that require energy. The energy is required to come in for that reaction to happen. An endergonic reaction. And just the opposite, exo always means out, or throwing things out, or releasing things to the outside world, or the outside of something, like an exoskeleton. An exergonic reaction is a reaction that releases energy out. And most of the time, what your body is going to do and what most cells are going to do is they're going to couple these two types of reactions to make things happen, right? You have one type of reaction, an exergonic one, that releases energy, and then an endergonic one is going to happen to use that energy. And that's going to happen over and over and over again. Okay? And this is all based on, in most cases, not all organisms, but the ones we're going to talk about, is based on ATP, which we talked about a little bit, but ATP is kind of what we call metabolic money. And it's called that because ATP is an energy storage form. Energy is stored in the bonds of this molecule. We're going to talk about how that's possible in just a second. But that energy is stored, and it can be spent, transferred, moved, sent somewhere else. It can be exchanged, traded. Okay, it's a physical molecule that holds energy. Kind of similar to how money works, right? Money is just a piece of paper that holds value. ATP is a molecule that holds energy. It carries it, and it can be moved from place to place. And so ATP is the molecule that most organisms use, or one of the most important molecules that most organisms use to exchange and move energy, to harvest it. And the way ATP works is ATP stands for, I don't know, oh, I do have it written down. ATP is adenosine triphosphate. And it is named that because it has an adenine base with a ribose sugar, which is adenosine. Now, you don't have to memorize that necessarily. You do have to memorize that it's an adenosine, which is the same, this adenine, which we'll talk more about later, is the same molecule that you see in DNA. We talked about that briefly in chapter 1, but we'll revisit in a couple chapters. So adenine is the same thing that you see in DNA. When you attach a sugar to it, a ribose, it becomes adenosine. Now this adenosine is called, or this ATP is called adenosine triphosphate, because wouldn't you know it, it's an adenosine with 3 phosphates attached. 1, 2, 3. There's 3 phosphates. Adenosine diphosphate is exactly the same, except it only has 2 phosphates. Okay, and adenosine monophosphate is the same thing, except it only has 1. Mono, di, tri. Okay, so the big thing about adenosine, and this is a weird concept when you think about it, is that these phosphate molecules are negatively charged. Ooh, I don't know what I just did. Okay. These adenosine molecules are negatively charged. Okay, and if you're not in my lab, that equal sign just means negative. Okay, it's just better than writing one line, because one line looks dumb. When you write it, you don't know what it means. The equal sign is another way to say negative. Okay, so these phosphate molecules are negatively charged. And some of you may remember this from high school chemistry or just wherever, but what happens when you put 2 of the same charge close together? They repel each other, okay? That's one of the bases of chemistry, that opposites attract. So if you have a positive and a negative charge, they want to be together. Negative charges, or 2 of the same charge, are going to push each other apart. Another really good way to think of this is magnets. So if you have a magnet, and we've all done this, you get those magnet sets and you try really hard to push the same side together and you can feel it pushing back on you because those two sides of the magnet, the same side, want to repel each other. Okay? Now I want you to think about those magnets and think about how it feels when you have two relatively strong magnets and you try to force them together on the same side. When you're doing that, it requires energy to hold them together, right? It requires your hands to be pushing them together for them to stay there. What happens if you were holding two magnets on a table that were pushing against each other and you let go? They're going to fly apart, right? The magnet's going to slide out of the way. Depending on how strong it is, it might shoot it out the other direction. That is what is happening in these molecules. So we call these two bonds between the phosphates, so this one right here and this one right here, they're what we call high energy bonds. Because what has basically happened is a cell has forced these two negative charges that don't want to be together and bound them together. It's handcuffed them together. And in that bond is the energy required to force them to be together. It's just like your two hands holding together two magnets. So while that bond is in place, there is potential energy there. There is energy that is stored in that bond. Because the second that bond is broken, the energy will be released. Exactly the same way as if you let go of a magnet, it's going to shoot off in the other direction. It's going to release the energy. So adenosine triphosphate has two, one right here, two right here, two high energy bonds. Now the high energy bonds are only the ones that are between phosphates. So this is something that trips people up a lot, is they think that because there's three phosphates, there's three high energy bonds. That's not true. There's two. It's the bonds that happen between the phosphates. So this bond over here is not between two phosphates. It's between a phosphate and an oxygen and a hydrogen. So there's not energy there. There are two high energy bonds in every molecule of ATP. And the way that most organisms and most cells use energy is they force together those phosphate molecules and store that energy in those bonds, and then break apart those bonds to release the energy when they need it. Okay, so this ADP, so taking adenosine diphosphate and adding on that says inorganic phosphate, it's just a phosphate. Okay, you can make ATP. And when you did this, this requires energy. Right? Because you're forcing these two negative charges who don't want to be together to be close to each other. It requires you to force them together. It requires energy. And that energy is stored in the bond. Okay? So when every time your body creates an ATP, it is storing two amounts of energy, two releases of energy. So if this requires energy to be put in, what kind of reaction is this? It's endergonic, right? So the creation of ATP is an endergonic reaction. It requires energy to be put in. Requires energy to go in. It is endergonic. Now, the opposite reaction is when you take ATP and you break off one of those phosphates. So you start with ATP and you break off a phosphate and you're left with ADP and inorganic phosphate. Okay. This releases energy. So if this reaction releases energy out, what type of reaction is this? Exergonic. Okay. And this is what cells do over and over and over again. They reuse these adenosines and these inorganic phosphates. They shove the phosphate on there to store the energy and then break the phosphate off to release the energy whenever they need it. Your body stores ATP in your organs and your muscles and your blood and everywhere else, and then when your body needs it, it breaks it down and releases that energy. Okay. So this is kind of just another view of that. This is the ATP/ADP cycle. Is that you have this adenosine, so this is an adenine, ribose, and triphosphate. So that's 3 phosphate groups. Notice we have 1, 2 high energy bonds. So this is ATP, ATP up here. If we break that phosphate off, we release the phosphate right here. That is going to release the energy. And this energy can then go on to do other reactions. Right? And what we're left with is adenine, ribose, and 2 phosphates, which is ADP. But if at any time the body is performing some type of digestion or energy harvesting, phosphates and energy will be absorbed, and we will create more ATP. And this is just what's going to happen over and over and over and over again. Right, I think I have. Okay, so this is just how this looks when you couple this ATP-ADP cycle into other types of reactions. And this is what happens in your body, okay? You couple the harvesting of energy to something that requires energy. So we kind of start up here. We're talking kind of again in terms of humans, but it's any organism that is capable of using glucose to harvest energy. So you have a glucose molecule, you have consumed it, you had some kind of food that had glucose in it. And this is what happens in your digestive tract and inside your body. So you consume something that has glucose in it. That glucose is broken down. Okay, breakdown is a breakdown catabolic or anabolic? Catabolic. So this is some type of catabolic reaction, and catabolic reactions release energy. So we have glucose breakdown, which releases energy, which also makes it exergonic. So this is both catabolic and exergonic, which are 2 different things. And what happens here is what you're left with is the products of this reaction, which end up being, you don't have to memorize this, but it's carbon dioxide, water, and heat. But the energy that is released from this breakdown of glucose, right? This is a catabolic reaction. It releases energy. That energy is then used to make ATP, to take this ADP, add energy to it, like we saw over here, add energy to it, and make ATP, which is an endergonic reaction because it requires energy. So you take the breakdown of this glucose, which is an exergonic reaction, and do it at the same time as an endergonic reaction. Now, this process in particular, you may have heard of before, you may not have. It's also known as the Krebs cycle. Sometimes it's called oxidative phosphorylation. Sometimes it's called aerobic respiration. This is a really, really complicated process that takes like 30 enzymes and stuff like that. I actually don't remember the numbers, but I think every glucose molecule, maybe you guys can remember, I think every glucose molecule produces something like 32 ATPs. And it requires like a dozen different enzymes. It's a very complicated process. The only time I ever got a B in college was when I had to memorize this for biochem, and I didn't do a good job. Okay, so the breakdown of glucose releases energy. That energy is then harvested to make ATP. And then this ATP that we have up here sits around until our body needs it. Maybe our body stores it in our liver. Maybe our body stores it in our heart. Maybe our body stores it in our brain. Wherever your body needs it, okay? Maybe it transfers it to another part of the body. But this ATP is a stable molecule that can then be stored, transferred, moved, exchanged, done anything with, okay? But when the body needs energy to do something, in this case, what it wants to do is make proteins from amino acids, right? Amino acids are the individual building block of a protein. So you have to hook together a bunch of amino acids to make a protein. That requires energy. It's endergonic. So what your body will then do, it needs energy. Your body says, I need to create this protein. I need to do something that requires this protein. I need to make it. In order to make it, I need energy. And so what it will do is it will take this ATP, break off one of those phosphates, which releases energy. So again, we're coupling an exergonic reaction, one that releases energy, with one that requires energy, an endergonic reaction. Okay? And this is what happens every time your body needs to do anything. And most things that your body does not require one ATP, it requires hundreds. So when you really think about how, I don't know, for me it's one of those things that's just really, it'll blow your mind if you think about it too hard. When you think about every time your body moves or breathes, how many hundreds of molecules of ATP you're using, and how many hundreds and thousands of enzymes are required to make that happen, and how delicate of a balance it is, and how everything is connected to everything else. It's just cool, to me anyway. All right, so, but this is, and again, keep in mind that this is a gross oversimplification of metabolism. Metabolism could be multiple classes all on its own, but this is all you really have to understand for our class, is that you couple an exergonic reaction with an endergonic one. You couple a reaction that releases energy with one that needs it. And that ATP is kind of the center molecule of that whole process. It's the money, it's the metabolic money that makes that happen. Okay? All right, so we are going to move on to chapter 8. We still have half an hour, so sorry to disappoint if you thought we were leaving. I cannot find my thing. Okay. So now we are going to move on to microbial genetics, right? And genetics and genes is a big couple of chapters. We'll probably spend at least 2 lectures going over this whole chapter. But genetics, what is genetics? And genes, what are they? So genetics is basically the subunit of how your body or any organism reproduces. Okay, so genetics is the passing of DNA from one organism, from one generation, we'll say, to the next. Okay, heredity is how DNA is passed from one organism to another, specifically one generation to the next. In other words, it's how any type of cell reproduces itself. Okay, at everything that exists in the world, their most base need or their most base purpose, I guess, and this could be kind of existential if you think about it too hard, but at your base, at every organism's base purpose is to make more of ourselves. Okay, when you think about bacteria or plants or humans or any other type of animal, one of our most base instincts is to make more copies of ourselves. And that applies to everybody. Okay, so heredity is how DNA is passed from one one organism, one generation to another. And then genetics is just the study of that heredity. Okay? So to understand how we pass genetic material between organisms, you first need to understand how DNA is replicated, and then how that DNA is then passed from organism to organism. And I just put this picture up here because it's kind of fun to think about. Heredity and genetics, they're weird, and they're very, very complicated. We as humans sexually reproduce. That's why, I have a son who looks half like me and half like my husband, right? His curls look exactly like mine. But now his hair color is the same as my husband's. He has thick hair like my husband, but his hair color is almost the same as mine. He has freckles like my mom, right? He's tall like his dad and my brothers. So there's a combination of DNA that comes from generations and generations and generations of humans before him, right? Genetics can be really complicated. It's the reason why two brown-haired people can have a redheaded kid, and there hasn't been a redhead in generations in their family. Or a black parent and a white parent can have a black baby or a white baby, right? Those things are weird. Genetics are interesting, and they're very cool, but they're very, very complicated. But at the base of all of that, on how I could have a kid that is going to be taller than me, almost certainly is going to be taller than me, is all based on how DNA replicates and how it gets passed from generation to generation, and how that is different between organisms who sexually reproduce and organisms who don't. Because remember that bacteria do not. They go through binary fission. They don't sexually reproduce. So we're going to talk about more about what sexual reproduction means and why it's different when bacteria reproduce versus like humans and animals. Okay? So first we got to talk about some just words that we have to define before we can actually talk about these processes. So chromosomes, which I'm sure is a word that you've heard before, but a chromosome is just a large structure of DNA and proteins. Okay, so DNA is the basics of the information. So this is the information or blueprint of how to make a cell, or a human, or an animal, or a plant. It is the blueprint or the information on how to build a whole other organism, whether that whole other organism is a single bacterial cell or an entire human. A chromosome is a large structure of that DNA, usually with some proteins involved. In humans, the proteins that are used are called histones. Those proteins are called histones. In other organisms, they're called other things. You're not going to have to remember them. But the way it works I can't remember, I thought I had pictures of it, but I guess not. I guess not. That's great. In humans, our DNA is wrapped around these histone proteins and makes those, I think we talked about this before. Maybe that's why I thought I had pictures of it. Those histone proteins make the DNA condense down into a very, very, very tiny, tiny, tiny structure, even though the amount of DNA in a chromosome is much bigger than you would expect. Okay? A gene, and this is just, again, another kind of word that we have to memorize. A gene is a small piece of DNA that contains the information for a specific product. Okay, and a gene can be any product. It can be the protein that gives your eyes color, or the protein that gives your skin the color that it is, or the protein that makes your hearts beat, or the protein that makes your kidneys work. Okay, it's any kind of protein, or it's any kind of product. But when we're looking at a specific piece of the DNA that codes for one product, that is a gene. And most of us have different versions of each of these genes. We all have the same basic genes, right? Most of us are born with eyeballs. Not everybody is, but most people are born with eyeballs. But all of our eyeballs are different colors. Even people who have blue eyes, I have blue eyes, but my blue eyes are more gray than my mom's. My mom's are more bright blue. My son's are more hazel, right? We all have different versions of the same thing, but there is a gene that codes for the protein that gives your eyes their color. We all just have different versions. Okay. So the genome, again, this is kind of just a vocabulary word we have to remember. The genome is an organism's entire collection of genetic material. And it will be all of their chromosomes if they have multiple. Remember that eukaryotes tend to have multiple chromosomes. The amount that they have is going to be different in every single species of organism. Humans have 46. We have 23 pairs. 23 that comes from our maternal parent and 23 that comes from our paternal parent. We have 46 total. Other animals and other plants and other things like that have different numbers, but most eukaryotes tend to have multiple chromosomes, and the genome is the collection of all of those chromosomes. Okay, so in my body, my genome would be my whole collection of my 46 chromosomes. Also remember that in eukaryotes, those chromosomes tend to be linear, like we showed over here, right? This is linear. This is a straight line. It's not a circle. Okay, so in eukaryotes, those chromosomes tend to be linear. And in prokaryotes, they tend to have a single chromosome, usually. And that single chromosome tends to be circular. All right, and then the last thing as a reminder is that in eukaryotes, where is the genetic material held? Inside their nucleus. And prokaryotes have no nucleus. Prokaryotes do not have a true nucleus. Eukaryotes have their genetic material inside of a nucleus. Prokaryotes don't. And then for viruses, we talked briefly about this before, but viruses tend to have DNA or RNA. Never both. Prokaryotes and eukaryotes have both. We'll talk about the purposes of both of those as we go through. Okay. Oops. Okay. So we kind of flew by this when we talked about it in chapter 1, but now we're going to take a closer look at the structure of DNA and how the structure of that DNA is going to affect how DNA is replicated. We have to understand the structure before we can understand how they make more copies. And I'm trying to remember what I wanted to say about this. There we go. The overall structure of a DNA molecule, we're mostly focused on DNA here, DNA structure. Remember, DNA is kind of our blueprint. We'll get to it in a little while, but remember that RNA is responsible mostly for making proteins. But DNA is mostly responsible as the blueprint. It's mostly involved in replication and reproduction. I think I talked about this more in a little while, but that's the overall purpose of DNA. DNA is mostly the information to build a new organism. It's what our body uses to build new cells, build new molecules, and reproduce itself. Okay, but a DNA is mostly, in all prokaryotes and eukaryotes, DNA is going to be a double helix, which means that you have two strands of DNA that wrap around each other. Sorry, my handwriting's getting out of control. But there's two strands of DNA that kind of wrap around each other. They form this coil-like molecule. There's two sides. You can see here, one of them is blue and one of them is red. Okay, but there's two molecules. Now, the key here is the difference between a covalent bond and a hydrogen bond. Now, a covalent bond, and we're going to talk about where you see each of these in this molecule, so you just got to kind of stick with me for a minute. A covalent bond is basically permanent. It is a very strong bond. They are very nearly permanent, and they are very difficult to break. They can be broken, but it's very difficult. Covalent bonds, very strong, very permanent. Hydrogen bonds, on the other hand, are weak and easily broken. Okay. Hydrogen bonds are weak and easily broken. Now, the key here is where you find covalent bonds versus where you find hydrogen bonds. And in all of the diagrams we're going to be looking at, I don't really know how to draw this, but the covalent bonds will be drawn as straight lines, and the hydrogen bonds will be represented by dashed lines to indicate that they're weak and easily broken. Okay, so this right here is looking at the structure of DNA kind of zoomed in, right? So we have two strands of DNA over here. This is if you zoomed in on those strands of DNA, okay? The two strands of DNA, so like this would be, we'll say like this one is the blue one. And this one is the red one, right? So if we're looking at this diagram over here, one strand is blue and one strand is red. We have two different strands of DNA. Notice where we have solid lines. So we have solid lines which indicate covalent bonds between the molecules of the individual strand. This is what's called, because these are very strong bonds, this is called a sugar phosphate backbone, because the basis of this strand is a sugar and a phosphate, right? You have the P is the phosphate, and then this is a sugar. Phosphate, sugar, and they're all connected, as you can see on this side too. You have a phosphate and a sugar connected by covalent bonds. So these backbones are called that because they're held together by very, very strong bonds. They're covalent, they're very nearly permanent. It's really hard to break a DNA strand. Okay, but where you have hydrogen bonds is down the middle where these bonds meet. Okay, so these are hydrogen bonds as they're listed right there. They are weak hydrogen bonds between the bases of the DNA molecule. So I know we talked about it back in chapter 1, but the DNA molecule has the sugar and the phosphate in the backbone, and then sticking out to the middle is the base. And those bases are the ones that are weakly attracted to each other with hydrogen bonds. Okay. So you have two strands, and those strands individually are very, very difficult to break. But they kind of interact with each other in the middle with hydrogen bonds. Okay, I kind of think about this as like somebody making a chain of humans, right? Has anybody ever played Red Rover? This was a game I used to play. It was super dangerous. I don't know if they stopped you guys from playing it. But Red Rover is a game where you hold hands. I have 25 cousins. We used to play this game all the time, and we all ended up with broken bones at some point, okay? So Red Rover is where you hold hands with a friend next to you, and one kid comes running at the group of people, and you try to break their hands apart, right? Really, it usually involves somebody getting clotheslined, but that's the game, right? Now, I kind of think about this as like the backbone would be the individual person, right? And the hydrogen bonds is where we're holding hands between us. If somebody ran full on into me, is my body gonna break in half? Probably not. Probably gonna get knocked over. but my body's not gonna break, okay? It's pretty hard to break a person's body, right? I have bones. Somebody running into me is not gonna split me in half, okay? Because I'm a whole person. I have lots of covalent bonds. However, if I was holding hands with somebody next to me, somebody running full force at just our hands, it's usually pretty easy to break that bond, right? Somebody running at it is gonna break our hands apart. That is kind of equivalent to a hydrogen bond versus a covalent bond. Okay, covalent bonds are stuck together. You have the two backbones, the two people standing next to each other, and them holding hands in between is the hydrogen bond. Okay? And that's how DNA works. You have these two strands of DNA, like two people standing next to each other to play Red Rover, and where they're holding hands in the middle is the hydrogen bonds. It's how they're kind of connected together, and that's how they form that DNA double helix. Okay, so again, those hydrogen bonds are very, very weak. They are going to be broken and put back together many, many, many times. Now, that's not to say that they're not important. The hydrogen bonds do hold the DNA in its structure. It's just important to note that that is a weak bond and it's easily broken. Now, DNA works in what we call Chargaff's rules. And I can't remember if we talked about this before, but if we did, we'll just talk about it again. And Chargaff's rules in DNA, we'll talk about something a little bit different when we get to RNA, but in DNA, adenine, or the A, is always hydrogen bound to thymine. Adenine and thymine like each other. They're always going to want to be next to each other. Okay, they're very attracted to each other. A and T will always be together. Okay, and if you look up here, right, we have an A and a T, and they're hydrogen bound. Adenine and thymine are always going to be hydrogen bound to each other. Always, always. Cytosine, which is a C, is always going to be bound to guanine, which is G. And keep in mind, this again is in DNA. When I say always, I mean always in DNA. We're going to talk about some other rules for RNA in a bit. But again, if you look up here, G is bound to C. G is bound to C. Okay, but adenine and thymine, again, remember adenine and thymine and cytosine and guanine are not long-term relationship people. These are weak bonds. They're attracted to each other, they like each other, but they're easily broken. It's like your weekend hookup, right? You're attracted to each other, you like each other, you want to hang out, but is this a strong, lifelong commitment? Probably not. Okay? Adenine and thymine are always going to be attracted to each other. Cytosine and guanine are always going to be attracted to each other in DNA. But again, they are easily broken. All right, you guys are never going to forget that now, are you? You're talking about weekend hookups? All right, so this is just another picture of DNA structure. This is what it looks like kind of zoomed out. You can see the DNA double helix with what's called the base pairs in the middle. And the base pair is where like the adenine and thymine are attracted to each other, and the cytosine and the guanine are attracted to each other. And then if you kind of zoom out, you can see this. This is the backbone here, and then the hydrogen bond base pairs in the middle. And then this is when you look at it even closer. Okay, just kind of zoomed in. Now, there's one more important thing that we have to talk about, and it's a little bit confusing. And again, we could get much more complicated about this, but I want you to take it at face value for what I say and try not to get too caught in the individual structures of things. Okay, because again, this can get way more complicated, but I'm trying to keep it as surface level as we possibly can while still getting the information that you need. The other thing about DNA that's important to note is DNA structure is what we call anti-parallel. All right, and I'm gonna explain what that means. So when you look at these individual DNA strands, and again, you don't have to memorize this structure, but what I want you to notice is this is one strand over here, and this is strand 2 over here, right? Look at where the oxygen is in this sugar. Okay, so here, this oxygen is at the top, okay? On this side, the oxygen is at the top. Over here, the oxygen is on the bottom. All right, so what this structure is, it's like I took two identical things, right? My hands are identical, two identical things, but before I attach them together, I flip one over, okay? And that is how DNA works. It's anti-parallel. It's two things that are identical, but one of them is mirrored and flipped upside down. And the way that we talk about this, and again, try not to get too caught up in the individual structures, is we label these sides of the DNA as either 3' or 5'. Now 3', when I say 3' out loud, you write that down as 3 with an apostrophe. Don't ask me why, I don't know why they decided that, but 3' is 3 with an apostrophe. And the other side is 5'. It's 5 with an apostrophe. And you can see, if you look at the structure, that this one has 5' at the top and 3' at the bottom. And this one has 3' at the top and 5' at the bottom. So they are just mirrored. One side is flipped over, and then that's how they're attracted to each other. Okay. All right, so you have like two hands, they're identical, but they're only gonna come together when one is flipped the other way. And that's what anti-parallel means. They're not completely parallel, you have to flip one over and make it opposite before they are attracted to each other. All right, and so try to remember 5' or 3'. Again, we could get more complicated with this, but just try to take it at surface level for now, okay? All right, I think we'll stop there. We'll do DNA replication next time.